this video, we're going to talk about the physiology of circulation and specifically microcirculation. So blood goes from the arterioles to the capillaries and then they go back to the veins. These are the component of the microcirculation. So look at this picture. Blood goes to the arterioles and there are some smooth muscle there, and then they will go to the meta-arterioles and then go to the capillary bed and then go back to the venules to go to the heart again. So meta-arterioles are when blood from arterioles goes directly to the venules without any exchange, so there will not go to the capillary bed. A smooth muscle cell in the arteriole wall can control the flow. We're gonna talk about it. And precapillaries sphincter provide local control, so sympathetic effect will not affect these precapillary sphincters. Capillaries are the functional unit. What do we mean by that? So they exchange nutrition, waste products, and fluid between the vascular and interstitial compartment. So, and the capillaries have only single layer, so when they just have single layer, it's easier to exchange. And not all capillaries are perfused all the time. For example, when your muscles are relaxed, there is no need for that capillaries inside your muscle to be perfused by the blood. A smooth muscle in the arteriolar wall and precapillaries sphincters are responsive to neurotransmitters, hormones, and metabolites. So endothelial cells, in response to shear stress from blood flow, they release vasodilator substance like prostacyclin and nitric oxide. So the effect will be change in total peripheral resistance. Now let's talk about this change. Autoregulation. So what is this autoregulation? In a rigid tube, when there is no elasticity, there will be a linear relationship between the flow and pressure. However, due to elasticity, we're gonna have something like this red line. But in autoregulation, these things does not happen, because, for example, when the pressure goes above some number, there should be, the flow shouldn't rise and there should be stay at the same level. And if the pressure goes down, there still should be some blood flow through that organ, and that is we call autoregulation, to keep the constant flow. And we explain it in kidney type vessel in our previous lecture. So the response of vascular smooth muscle is compensatory regulation. What makes this autoregulation? There will be a vasomotion. What do we mean by that? So a smooth muscle in arterioles undergoes cyclic contraction and relaxation, and this will be called oscillation, exactly like how worm moved inside on the ground. And what is the mechanism? Cyclic change of calcium signals, they cause synchronized smooth muscle contraction, and these contractions improve the flow. So more efficient oscillation prevent latch states, which is common in a smooth muscle because of having low level of ATP. So what is plug and bolus flow? So look at this graph. As you can see, we have one red blood cell, then some plasma, then another red blood cell, then plasma. You can see plasma and red blood cell in same place. And as you can see, red blood cell and white blood cell are bigger than capillaries, and they need some deformation to be able to pass. And white blood cell are bigger than red blood cell. So capillaries have a smaller diameter than red blood cell. Red blood cells should undergo severe deformation while they travel through the capillaries, and the donut shape of red blood cell make it more flexible. Each red blood cell is like a plug followed by a plasma, which is like a bolus between another red blood cell. This mechanism helps better exchange. Red blood cell does not ever come into contact with the wall because of the wall's glycocalyx layer, which cover the internal surface of the endothelium. So impact of white blood cell on flow in microcirculation. As you know, the size of white blood cells are bigger than red blood cells, and number of red blood cells are more than number of white blood cells. So white blood cell still can pass through the capillary, but with slower flow. And frequently, these white blood cells make a block in the capillary that must build a pressure gradient from the heart to push it through the capillaries. In normal number of the white blood cell, they don't make any problem. However, in some diseases that the white blood cell count goes up, for example blood cancers, they create a problem, and that patient's spleen, one of the organs with high number of capillaries, get bigger than normal situation. Exchange of substance across the capillary walls. So we have three different type of capillaries: continuous capillaries and fenestrated capillaries and discontinued capillaries, and they are divided to the three groups by the distance between two cells. In continuous, the capillary cells and the endothelial cell are very tight together, and they only pass very specific things like we have in brain, we have in fat cells, we have in muscles and nerve cells. But like fenestrated capillaries, they allow more molecules to pass, and they are very good for absorption. And discontinuous capillary is when there is a filtration. For example, inside the liver, when there need to be some filter, or in bone marrow because of making many cells. And in discontinuous capillary, some cells can pass through the capillaries, as you can have it in a spleen or bone marrow. So exchange that is happening between the between the inside the structure of the capillary and interstitial fluid is through the simple diffusion. Some of them go through the endothelial cell, and some of them goes between the endothelial cell, and we call it two different pathways. So transcellular pathway through the endothelial cell, and lipid soluble substance can pass like oxygen and carbon dioxide. And paracellular pathway is for water soluble substance like water, ions and glucose. In paracellular pathway, substance travel in aqueous cleft between the endothelial cell, and protein are usually unable to pass in the paracellular pathway. In the brain, these clefts that we explained are very tight, so no protein can pass through blood-brain barrier because they need to have some security there. It's somewhere very specific. Capillaries allow protein to cross through the pinocyte vesicle, as you study in your biology. Thank you for watching my video. Don't forget to share this with your friends, and please subscribe to XMed Education and like and share our video. Thank you very much. Have a nice day and goodbye.